Today we're going to talk about interplanetary transfer. This is basically the process of how material can go from one planet to the other. This defines a lot of how life on Earth originated, maybe through deposits from Mars. And in this process and in this video, we're going to look at how living organisms inside rocks could have survived this trip. Now, a more scientific term is planetary interchange of bioactive material. And with this, we have an equation that can quantify the potentials for a living organism to survive this entire process alone. Now, throughout this video, you're going to see a lot of Bacillus subtilis, which is used in a lot of experiments as a living organism, and it's very resistant to heat, ultraviolet radiation, desiccation, vacuum-like areas, lysosomes, chemicals, which is organic solvents and oxidizing agents. Number 1. The ejection from the donor planet. This occurs when something strikes a planet and we have deposits that come off of its surface, and this is the first step. Now, in order to test this, the jerk and the acceleration, scientists and researchers got together and they put spores on the end of a bullet and they shot it out to see exactly what the survival rate was. They really wanted to see whether acceleration and jerk would be a main issue and an impediment in the process. And it turns out it's not. 82% survived. Now, in order to test the shock and heating in the absence of acceleration, researchers took multiple foam core layers and shot a pellet all the way down into a target to see exactly what happened. The interesting part was spallation occurred at the blast zone, where fragments actually go into other layers that are above it in recoil. Now, the observations taken from all of this was really that we had the first simulation of a launch by spallation, the spores were able to tolerate acceleration and jerk, and the shock and heating are more important factors. The spores in the granite survived spallation from the launch, and lightly shocked spallation fragments occurred. This was mainly successful. Number 2, we look at the transit through space. This is long travels through space, through a vacuum, extreme desiccation, radiation, temperature extremes, and this is to see if organisms could actually survive this trip. Now, researchers were able to test this process through the space station, where they took the Bacillus subtilis and put on the outside exterior part. And what this did was it subjected the Bacillus subtilis to conditions of space and what those conditions might be. And they tried to simulate the Mars environment, the Earth environment, and the in-between. Now, looking further into this experiment, we see that multiple layers are collapsed on top of each other, which involve a filter O-ring, an AI disc with spores, and subsequent layers of spores. Now, this creates variability in the sample, and that the ones at the top are more exposed to the sun, whereas the ones at the bottom are protected from solar radiation. Now, the conclusion for this experiment was that over 100 genes were upregulated, the space exposure caused more damage than did Mars environment. Upon arrival to Mars, potential forward contamination would have occurred, and the characterization of individual gene candidates is still in progress. Now, for number 3, we look at the entry onto the recipient planet. The major concerns involved in this is heating, if that present planet has its own atmosphere, and also the impact. The material is going to have to hit the surface in some sort of way, and we want to test whether the living organisms could actually survive that type of heating and that type of impact into another host planet. Now, in order to test this process, researchers took a missile with B subtilis attached to a piece of granite and put different layers of spores onto the front, the back, the left, the right, and the top. And it turns out the ones in the back actually had the greatest amount of survival. Simply enough, the missile was shot up into the air, 127 kilometers high, and landed back on the Earth's surface. From there, we were able to collect the results and see what happened. Now, in conclusion from this entire experiment, we find that surface rocks can be exchanged between Earth and Mars via impacts. The Earth's surface rocks contain hardy endolithic bacteria, and microorganisms can survive the launch, spaceflight, and atmospheric entry environments. And thus concludes our video on interplanetary transfer. I hope by now that you understand that we could actually be organisms from an entirely different planet, and we don't even know it. I hope this video was helpful for you in understanding the process, and thanks for watching.